The eye. The eye is a complex sensory organ that provides the sense of sight. In many ways, the eye is similar to a digital camera. Like the optical system of a camera, the cornea and lens of the eye capture and automatically focus light. The iris also automatically adjusts the eye to difference of illumination of visual fields. In many aspects, the optical system of the eye is far more elaborate and complex than a camera. For example, the eye has the ability to track moving objects with coordinated eye movements. The eye can also protect, maintain, self-repair, and clean its transparent optical system. The wall of the eye consists of three concentric layers or coats. The eyeball is composed of three structural layers. The corneoscleral coat, the outer a fibrous layer, includes the sclera, the white portion, and the cornea, the transparent portion. The vascular coat has the middle layer or uvea, includes the choroid and the stroma of the ciliary body and iris. The retina, the inner layer, includes an outer pigment epithelium, the inner neural retina, and the epithelium of the ciliary body and iris. The neural retina is continuous with the central nervous system through the optic nerve. The chambers of the eye. The layers of the eye and the lens serve as boundaries for three chambers within the eye. The chambers of the eye are the following. The anterior chamber is the space between the cornea and the iris. The posterior chamber is the space between posterior surface of the iris and the anterior surface of the lens. The vitreous chamber is the space between the posterior surface of the lens and the neural retina. The cornea consists of 5 layers, 3 cellular layers and 2 non-cellular layers. It consists of 3 cellular layers that are distinct in both appearance and origin. These layers are separated by 2 important membranes that appear homogeneous when viewed in the light microscope. Thus, the 5 layers of the cornea seen in a transverse section are the following: corneal epithelium, Bowman's membrane, corneoscleral stroma, Descemet membrane, corneal endothelium. This electron micrograph shows parts of 3 lamellae and a portion of a corneal fibroblast between 2 of the lamellae. Note that the collagen fibrils in adjacent lamellae are oriented at right angles to one another. This photomicrograph of the human eye shows the anterior portion of the ciliary body and parts of the iris and sclera. The inner surface of the ciliary body forms radially arranged, right-shaped elevations, the ciliary processes, to which the sonnular fibers are anchored. The ciliary body contains the ciliary muscle, connective tissue with blood vessels of the vascular coat, and the ciliary epithelium, which is responsible for the production of aqueous humor. Anterior to the ciliary body, between the iris and the cornea, is the iridocorneal angle, the scleral venous sinus, the canal of Schlemm, is located in the close proximity to this angle and drains the arcus humor to regulate intraocular pressure. This schematic diagram shows the layer of the iris. Note that the pigmented epithelial cells are reflected as occurs at the pupillary margin of the iris. The two layers of pigmented epithelial cells are in contact with the dilator pupillae muscle. The incomplete layer of fibroblasts and stromal melanocytes is indicated on the anterior surface of the iris. On the basis of histologic features that are evident in the photomicrograph on right, the retina can be divided into 10 layers. The layers correspond to the diagram on left, which shows the distribution of the major cells of the retina. Note that light enters the retina and passes through its inner layers before reaching the photoreceptor of the rods and cones that are closely associated with retinal pigment epithelium. Also, the interrelationship between the bipolar neurons and ganglion cells that carry electrical impulse from the retina to the brain is clearly visible. The Bruch membrane separates the inner layer of the vascular coat from the retinal pigment epithelium. The first electron micrograph shows the junction between the inner and outer segments of the rod cell. The outer segments contains the horizontally flattened discs. The plane of this section passes through the connecting stalk and cilium. A central cilium and its basal body and a calyceal process are identified. Another electron micrograph shows a similar section of a cone cell. The interior of the discs in this outer segment of the cone is continuous with the extracellular space. Retinal pigment epithelial cells contain numerous elongated melanin granules that are aggregated in the apical portion of the cell, where the microvilli extend from the surface toward the outer segments of the rod and cone cells. The retinal pigment epithelial cells contain numerous mitochondria and phagosomes. The arrow indicates the location of the junctional complex between two adjacent cells. This is a normal view of the fundus in ophthalmoscopic examination of the right eye. The site where the axons converge to form the optic nerve is called the optic disc, because the optic disc is devoid of photoreceptor cells. It is a blind spot in the visual field. From the center of the optic nerve, central retinal vessels emerge. The artery divides into upper and lower branch, each of which further divides into nasal and temporal branch. Veins have a similar pattern of tributaries, approximately 17 or 2.5 times optic disc diameters lateral to the disc. The slightly oval shaped, blood vessel free and pigmented area represents the macula lutea. The fovea centralis, a shallow depression in the center of the macula lutea, is also visible. Thanks, eye, for let me see not only the things that I want to see, but all the things that I deserve to see. This was the eye.